Good. So first of all, let me thank organizers, Michael and Johannes, for inviting me to that amazing, nice workshop. I'm really enjoying the topics here. And thanks to Johannes and Florian to shift the program. I'm a proud father of an amazing daughter, and she was just defending her master thesis, actually in Denmark, Michael. And you know my daughter because when we worked together in Konstanz, she was just born. And now she's defending the master thesis in Denmark, actually on NV center magnetometry in biological samples. So I had to attend this. So thanks for making this possible. Okay, yes, so I will talk about the observation of a molecular bond between an ion and Rydberg atoms using a pulsed ion microscope. And this is work done in Stuttgart in this group, also an amazing group actually. Florian is here, but there's also Robert and then Nico Zuber, who is the leading senior PhD student who was really building this microscope for a long time, together with Christian here, Veit, who has already graduated some time ago, and then there are other team members, Virat, Moritz, and Ichen, and also Oscar, actually from Costa Rica. So this talk is going to be an experimental talk, so I start with the experimental device without any further motivation. I will introduce this ion microscope to you and then talk about the first application of that microscope in the following. So that's the device. Yeah, it's a cold atom machine. Oh, okay, okay. Yes, so that's the machine. Yes, it's a cold atom machine, a normal one with a Siemens lower and a science chamber, and then there is this huge device on top of this machine, which is our ion microscope. Yes, so it's able to detect single ions, and it's a pulsed ion microscope, which means that we can switch it on a time scale much faster than 100 nanoseconds, actually tens of nanoseconds, and extract whatever is ion-like, yes, anything that has a charge, up to this detector at the top. And that detector at the top, oops, not switching. Let's switch it manually, maybe. Yeah. Is an MCP detector that has spatial and temporal resolution. Yes, many of you are using this. Yeah. And it has a delay line detector, so it gives very good time information as well. And in between, we have three different lens elements for ions, which are just cylindrical elements of electric field carrying electrodes. Yes, one of these lenses is placed here, another one here, and a third one down here, right there where we have our cold atoms. So the cold atoms are going to sit in this place here, and the three lenses are built such that we can magnify anything that happens in real space in this cloud of atoms by a factor of 1,500. Okay, which together with the resolution, the spatial resolution of this MCP results in a spatial resolution of 200 nanometers or better. Yes. So it's better than an optical microscope, for example. So that's a close-up, yes, and the atoms are going to sit in here. And in this little, let's say, tin can, yes, we can also, and that's very important, compensate the electric field to a very high accuracy. And it turns out that if you use this imaging capability of that ion microscope, you can watch the movement of the ion and use that observation to compensate any stray electric field that you might have in your chamber. Although we try to shield the whole volume as good as we can from stray fields. Of course, there are finite stray fields. And in previous experiments that some of you know, we have always used Rydberg spectroscopy, measuring Stark maps at very high ends to compensate the electric field as good as we can. But observing the motion of a single ion that is just dragged along by a stray field is much better than that, actually 1 order of magnitude better. So we can now in that tin can have a field compensation down to 100 microvolts per centimeter, which gives us time to observe this floating single ion for 100 microseconds or longer in the field of view of our microscope. And at an instant in time when we want to observe it, we switch on the microscope, which involves kilovolt voltages. Yes, so we switch this on in tens of nanoseconds and image the ions. And then, of course, we have to go back to zero field, and the experiment is always repeating between absolute zero and very large fields to extract the ions. So that sort of was the experimental challenge to solve here. Yeah, I think it's a masterpiece, not so mostly of our workshop again. I mean, you mentioned the technicians, and that is what I also want to do here. These mechanical elements of these lenses have to be machined with a 50 micrometer tolerance. That's something that our mechanical workshop cannot even measure whether it's that good. And also the electric fields that we are applying at the high voltages have to be good to 10 to the minus 6 or something like this. So we need very quiet high voltage power supplies that we can also switch very fast. So that was also done in our electronics workshop. Okay, so what can we do with this now? Yes, imagine you have something interesting. Yeah, it's doing something that you want to observe, and then at some point you can just switch on your microscope and you observe it. Yes, here you see another example of this. So that's the way we view the operation of our microscope. Yes, it's a pulsed one. That's very important. So whatever happens down here is in the cold cloud. Yeah, let's say you put a lattice or something, some cold ensemble, starts at T1. Yes, where we, for example, excite a Rydberg state or an ion by photoionization. Then we can wait for a certain time until we want to look at it. Then at T2, we switch on the microscope, we extract everything, and we read out after a certain time of flight in this microscope at T3, what is the coordinate X and Y. Yes. So this time of flight information is actually also important because it allows you, for example, to distinguish in the future, for example, various isotopes. Yes. Or Rydberg atoms from ions, as we will see. Okay, and yeah, so this is just summarizing this device, spatial resolution 200 nanometers or less, time resolution certainly much better than 100 nanoseconds. This time information is actually able to give you information about the third dimension. Yes, so in principle, it's a 3D imaging device. Okay, good. Then the next ingredient that I have to remind you, I'm sure all of you know this, is the Stark map of rubidium atoms that we are going to use in that experiment. And the main feature that you should just remember is that if we excite an S state or a P state or a D state, they get sucked into high electric fields. They are high field seekers. But if you look at the hydrogenic manifold up here, there are also some low field seekers, which is going to be important to understand the molecular bond. So if we first just excite the atoms, in this case, to the 129 S state, yes, around an ion. So what do? We first excite an ion. It's floating, yes. Fields are absolutely zeroed out. The ion is there, we can observe it, and then we just excite a second atom to a Rydberg state. Yes, so what we are looking at now are the relative spatial coordinates between the ion that we have produced and the Rydberg atom. The Rydberg atom are these blue counts here, and we are always centering all the spatial information around the ion. So we are just plotting the relative coordinates between the ion and the Rydberg atom. Yes. So if you remember now, yes, there's an attractive force between the ion and the Rydberg atom because of that Stark map. Yes, the S state sort of has a polarizability, and gets sucked in on this 1/R4 potential. And you can map out that potential by just changing the excitation laser detuning. Yes, here 3 megahertz, 6 megahertz, 9 and 15 megahertz. And you see that the cloud of Rydberg atoms around that ion basically just shrinks. And if you plot now the distribution of atoms, you integrate the radial part out because that's not very important here. That the angular, sorry, you integrate the angular part out because that's not very important, because you know that is just dominated by the finite size of our optical dipole trap. Then you get these histograms, these blue-coded histograms here, and you can compare that with the potential energy curve of that 129S state in the vicinity of that ion, and you see that you can map out that potential energy curve relatively nicely by just changing the laser detuning here on this axis. The excitation follows that potential energy curve. Actually, you can also now look at dynamics. If you excite these atoms somewhere at some distance and you just wait, you can see how the Rydberg atoms get sucked into the ion. Yes, and you see there's an interesting dynamics. And I think Peter, you see, he will maybe talk about some theory to explain this behavior tomorrow. But for now, let's move on to that Rydberg molecule that was proposed by Johannes and the Reitel group, or Johannes group and the Reitel group, that, you know, in contrast to what we just saw, don't look at the S state, yeah, which is just sucked into high fields, but look at the P state here, which is first sucked in, but then crosses with the low field seeking high L states of the hydrogenic manifold. So we are going to look now at this kind of position here, where you can expect that there could be something like bound states. Yes. If you look at this Stark map only, it's not so clear, you know, how exactly this potential will look like. And you have to remember, and that was what is done by Johannes, that the Rydberg atom is a finite size object. And the field around the ion on that scale is very inhomogeneous. So you cannot just convert that Stark map of a Rydberg atom in a homogeneous field into a potential energy surface. You have to do something more. And what you need to do is to look at the higher multipoles, yes, of that charge distribution, if you like, to calculate really how the potential energy surface around that crossing point actually looks like. Yes. So these are the two references here where this was done theoretically. And now if you look at this crossing, in this case as a function of distance now, it's the energy as a function of distance around that crossing of this P state here, then you see that eventually here, you know, if you zoom in even more, there's a harmonic oscillator potential. Yes. And you can calculate the vibrational frequency. In this particular case, it's 11 megahertz. Yeah. So, and 11 megahertz is something that you can nicely spectroscopically resolve. Yeah. So we said, okay, let's use our microscope and look at it, and how does it look like in real space? Okay. So if you think about this harmonic oscillator and you think about the binding mechanism, yes, then what you see is if you calculate the electronic eigenstates on the right side of the potential, then the electrons are on the left side. On the other side, that's the low field seeking, the charge is on the other side. So the dipole moment of the Rydberg atom is flipping back and forth as you go through the minimum of that harmonic oscillators. In that sense, I would say it's a binding mechanism that you don't find in a normal physical chemistry textbook. Yes. Okay. So we call it the flipping dipole interaction. Yes, which was nicely discussed in these theory papers. So let's look at the experimental sequence. We first create an ion. Yeah, the way we do this is just photoionization of rubidium. Yeah, just the 2-photon excitation, just barely above the continuum. then we have a very cold ion, yes, and it's almost not moving. And then in the second pulse, we excite, again with two-photon excitation, a Rydberg atom, but we are choosing the tuning such that we are hitting one of these vibrational levels, yes. Now you realize that this is a two-photon excitation from an S state, but the main component of the final state is a P state, so it should be forbidden, yes. But due to the mixture of this interaction with all the multipoles around that minimum, there is also P and D state contributions, which allows us, nevertheless, to excite this atom just with two photons, this Rydberg atom. Yeah. Then we wait, and then we actually switch on first a small electric field to work on the ion and extract that ion, basically break the bond, and only, to first order, only move the ion towards the detector, and then a little bit later field ionize the Rydberg atom such that the electron is gone. And then we have two ions, one the original ion, and the other one is the field ionized Rydberg atom. But as one of them has a head start, yes, we can distinguish them then on the MCP detector. That's actually real data. These are the two arrival time peaks that we are seeing on the MCP detector. So we can tell, okay, this one was a Rydberg atom and this one was an ion. Okay. So then we can do first the spectroscopy. That is just using the frequency resolution, which gives these nice vibrational states that we can nicely resolve and compare to theory. The gray lines are the calculated positions of these stationary states, and you can even see that this other bound state down here, one or two bound states in this lower branch, are also present in our measurement here. Yes. And then we looked at these objects first as we are dragging them along due to a finite electric field. So we do a very poor man's version of mass spectrometry just to convince ourselves that ions that started out in this red area that are dragged along in this electric field to the upper right corner as single ions are slowed down as soon as they are attached to another Rydberg atom, because this is just doubling the mass. Yeah? So if you apply a force, the molecules will just move half the distance. Yes. So this tells you at 2.3 millivolts per centimeter, the bond is still existing, and the ion is dragging the Rydberg atom behind. That's the blue signal here. Yes. So this is a bound system, yes, which you can observe in real space with your microscope. But now we can also look at the shape of the molecule, yes, because in this case, the 69P state has a bond length of about 4 micrometers, which we can actually measure to be 4.3 micrometers. And we can see how the molecule looks like. Now, interestingly, it is not round. Yeah, that was a surprise for us at first. Because we thought, okay, the electric field is round, yeah, so I mean, all this physics should be totally isotropic. So why do we see a molecule? This is the experimental data up here that shows what is called an alignment, yeah, preferred direction somehow. Yes. Now, after thinking about it a little bit and doing some calculations, it became clear that it has to do with the polarizations of the exciting lasers. If you take this into account, you know, all the Clebsch-Gordon gymnastics in a frame that is sort of pointing in any direction around the ion, then you see that the excitation probability due to polarized two-photon excitation actually is leading to that alignment, yes, that you're actually observing here. So, and you can change the polarization from here to here. Yes, you rotate the linear polarization, then the distribution changes, and you can explain it nicely by just calculating those matrix elements for all these orientations. Okay, so that's the first application of that spatially resolving microscope. Tells you that your molecule is actually already aligned by the excitation process. Okay, and finally, let's look at some dynamics and also think about what this molecule actually does if you apply an electric field. Here's the molecular potential energy surface with a part on the left side of the ion and a part on the right side of the ion. Now, if you are applying an electric field, then several things happen. Yes, you are sort of polarizing this molecular ion in a sense that this the whole Rydberg cloud is moving in one direction. Yes, on the right side, the minimum of the potential is moving to the right. And on the left side, it's also moving to the right. But what is also happening is that the two energy minima are changing. Yes, one is going down on this side because the electric field adds up, and here it is subtracted. So eventually, here the excitation requires a higher energy. On this side, it requires a lower energy. So if you now excite with a fixed detuning, delta here, you can arrange the excitation such that you only excite on the left side, yes? Which is resulting in what is called in molecular physics an orientation, yes? So as a function of the detuning, you can plot that orientation, which means in real space, the excitation of Rydberg atoms now happens only on one side of the ion and not on the other side. Yes. So this way, so you see the zero field reference is here. So that was the spatially resolved molecule, how it looks in zero field. Now you apply 5 or 10 millivolts, and then at a negative detuning, as explained here, you see that there is a, let's call it a wave packet, yes, that is prepared on one side of the ions. Okay. And you can understand that if you are increasing the electric field, then the maximum actually moves to larger negative detunings. So that's, if you like, a sort of Stark shift measurement. But what is more interesting is that if you now take this 10 millivolts and switch this off again, then what happens is that if you have excited your wave packet on this left side here, you are going back to the original situation, but if you do it fast enough, we can do it in 10 nanoseconds, yeah, then this wave packet is projected on the old potential energy surface, and it will start to oscillate, yes. So here you can see the radial evolution of the wave packet. It's a little bit hard to see because there's a lot of more details that are going on. If we are just looking at the center position of the distribution, the spatial distribution of this wave packet next to the ion, then you see that we can, whoops, where is my thing here? Oops, here. I have to cut it back down here, yeah. Then you can see that we can observe the vibrational oscillation of that Rydberg atom around this minimum of that potential energy surface. And you see maybe one or two excitations here, oscillations. Yes. Everything happens, by the way, on the time scale of 1 microsecond. Yeah? So it's important that this microscope has this kind of resolution on the scale of hundreds of nanoseconds or faster. And you see that the amplitude that we are observing here is on the order of a few hundred nanometers. Yes. Okay. So if you want to understand that, that's actually work in progress. This is the experimental data of that vibrational wave packet dynamics. Then you can do a classical simulation, yes, which just takes this potential energy surface, and in this particular case, just takes the ground state of vibration and puts it there. Then you see it's okay, but it's not perfect yet. Yes, so we still have to work on this. And we have to remember that it's actually a three-dimensional problem, including all the rotational degrees of freedom that are not taken into account properly here, and also that we are exciting not only the ground state, but the higher excited states. And so we still have to work on the theory here to make it fit the experiment a little bit better with an extended model. So this is just a preliminary version, but you can already see that, you know, you get reasonable agreement. Okay, so this is summarizing the talk. I showed you how we are using this pulsed microscope to observe that new molecular ion between a single ion that we can prepare, and a single Rydberg atom that we are exciting in the vicinity. We can prepare wave packets to study the vibrational dynamics in real space and in real time. And of course, you can think about many more things now. You can excite two Rydberg atoms. Let's say think about a trimer and ask yourself about the correlations, because this is it's a large molecule, but the Rydberg atoms also have strong interactions, so there might be correlations in that three-body system. But you can also use that microscope for different purposes that were also discussed by Georg. I don't know where he is now. Here he is, yes. I mean, you have seen very nicely in his talk Friedel oscillations, polaron physics around an ion, and so on. And the spatial resolution of that microscope is designed such that, you know, it should be able to detect these things in real space. Okay, so with this, I thank you for your attention, and also in our group, we are looking for postdocs and PhD students. Thank you.
Thank you, Tilman, for a very nice talk. Tilman, I will the first question. This vibrational wave packet is not like a quantum pendulum. You have the releases of a quantum pendulum or not?
I would, at the moment, more think about this in classical terms. Yes. Yes, the lifetime is on the order of 10 microseconds. And we have not investigated very systematically. We would expect from first principles a longer lifetime. What we are suspecting is that around these minima here in the potential energy surface, there are a lot of other states. Actually, here you have seen it. Yes, here. There are a lot of other states, and all the states involved have a very large electric dipole moment. And there are very large electric dipole transitions that might be driven by spurious radio frequency fields in the lab and so on. And if I would have to bet, I would bet on just technical noise in our setup that is allowing for, you know, transitions from that bound state, let's say this one, to other states here. But we have not systematically studied this. The measured lifetime is 10 microseconds, or actually 7 or so. Yes.
One question, what's the origin, the physical origin of the damping there, of the oscillation of the wave packet?
Yes, so this is a three-dimensional problem. So maybe I should go back to this picture here. You see the initial state is a wave packet like this. It's not a quantum mechanical state, yes, it's not the lowest possible state and this quantum mechanical wave packet, but I would say more a thermal wave packet, yes, that is just sitting on one side of the ion here, yes. And now if you let the potential relax back to its original position, what happens is that there is a radial oscillation, but there's also some dynamics going on in the angular degree of freedom, yes. And what we are plotting now is only the radial part, yes, but they are coupled together, yes. And so that leads to an efficient damping of these oscillations after two oscillations, as you have seen. And that's actually also reproduced in our classical simulation of that problem using a 3D potential energy surface.
Yeah, thanks so much for these great results. I was wondering, these potentials are adiabatic potentials, I think, right? So yes. If you drive these time-dependent vibrational wave packets fast enough, could you see non-adiabatic transitions to neighboring potentials?
Yes, I think there are even calculations, at least by the Reitel group. Maybe Johannes, you have also done some calculations. Whoops, where is it? Whoops. The gap basically to the next level here, this is about 150 megahertz. Yeah, so the 11 megahertz is slow on that scale. Yeah, so that's why we can see a few oscillations, but eventually there might be a limitation due to these transitions. Yeah.
So thank you, Tilman, for the beautiful talk. you've shown that when E equals 0, the polarization of the laser breaks the spherical symmetry, but, well, maybe there's a simple answer. You spectrally choose the eigenstates to which you excite, and if the eigenstates are spherically symmetric, I would expect maybe the matrix element to be lower, but then the state you populate still to be spherically symmetric, no?
So I'm not sure whether I understand the question fully, but the rotational constant is extremely small here, yes? So this is basically, so if you talk about the eigenstates that have no spherical symmetries, then you probably talk about the rotational eigenstates, right? But as the binding energy is 4 micrometers, you know, this rotational constant is less than a kilohertz or so, yes? So not anything we can resolve here.
So you
yes?
have nearly degenerate
rotational states, and therefore it's fully classical in the rotational degree of freedom. And the only thing that counts then in the end are these matrix elements in the Stark map, yes, that are polarization dependent.
Yeah. Hi.
Hey.
So great talk. I had a technical question about the microscope.
Yes.
So you showed in a small time of flight. Yes, just previous. Yes, that one. So you show 2 hits on the MCP, which are from a Rydberg originated ion and the direct ion.
Yes.
And there's a timing difference between the two. But if you, I mean, from the cartoons of your microscope, you are doing a lot of lensing. So how can you actually determine which one is which? Because you are going through a whole bunch of
well, I mean, you can do the
And sorry, so since you are accumulating data over multiple instances, you are not going to be able to, I mean, can you still do this?
Yeah, so I mean, if you just average all the data, then you see 2 very clear and distinct peaks, yes? So if this is your concern about drifts or so, that's not a problem. And if you think about it, it's actually, you know, you first accelerate the ion in a low field a little bit towards the detector, which is here on the right side, and then you field ionize the Rydberg atom, yes? And then you have a huge electric field, a huge slope, and that means actually that the Rydberg atom ion, yes, will overtake the other one as it flies through the microscope, yes? That's what we observe, yes? You just take the ion alone, and then you look at the Rydberg atom signal, and it appears on the other side, sort of, yes? It overtakes, yes? It's quite interesting, yeah? But that's, of course, expected from classical simulations, yes?
Yeah, I enjoyed also the new results, from the last part of the talk. So there, of course, you have to be, you accelerate the ion while you excite this molecular state. And of course, probably you have to do this very gently.
Yes.
So what are the velocities, or how do you make sure that the ion doesn't get so fast that the relative velocity between atom and ion is so large that it will not work anymore?
You hit the point. So if you look at the amplitude of the oscillation here, it is actually much smaller than the amplitude that we observe later on in the experiment. And that has exactly to do with this pulse sequence that we are using, that if we are extracting the ion first, it sort of drags the Rydberg atom along a little bit, yes? And in this measurement, it actually helps us to magnify the amplitude. Yes, so I mean, I told you that we are measuring here 200 nanometers amplitude or something like this, but the original amplitude in that potential is actually much smaller, about a factor of 10 smaller. And so by that pulse sequence, we get an amplification factor of that amplitude because of that dragging here, yes, which is pretty funny, yes, unexpected also, yes.
Okay, let's close the discussion here. Let's thank Florian, Johannes, and Tilman again.